All right, let's begin. Chapter 16, molecular basis of inheritance. But seeing as how we also have exam 3 starting tomorrow, and this is the last chance to talk about exam 3 stuff, besides Chase's super session right after this. Also just going to be open Q&A about anything exam 3, which I think is 14 through 16, right? Should be 14 through 16. So there's your memes. Hope you enjoyed them. I like the little one on the left because that's very much me and my wife. I brought home a couple of Kit Kats, and I come home and they're all gone. And I was like, all the Kit Kats are gone. And she's like, well, I ate them all. I'm like, well, but I wanted one. And she's like, well, Kit Kats are my favorite candy bar. And I'm like, they're also my favorite candy bar. And I brought home like 4. So it was a good time. All right, so we're going to jump straight into it. These are the questions that I got in emails and Canvas messages and whatnot today. Once these are through, I'll kind of just open up to the rest of the questions. That way, if you have a question that gets answered, then we've already taken care of it. And then we'll go through the material from today, depending on just how much time we have left, right? Mostly we're just trying to answer your questions that you have today. So, someone always sticks the pen here. At least I always know where it is, right? All right, so this huge question. Assume that there is a gene in apples that determines fruit color and a second gene that determines fruit size. Let A represent dominant allele for big apples and represent the recessive allele for small apples with a little a. Similarly, let big R represent the dominant allele for red apples and little r represent recessive allele for yellow apples. You have one tree that produces big yellow and another tree that produces small red. When the two are crossed, you find that half the new trees produce big red and half produce big yellow. What are the genotypes of the parents? Okay, so these are the alleles that we're dealing with. First off, tell me as much as you know already about the parent genotypes. You can logically deduce some of the genotype just based off of what I've told you about the parents so far. Don't even worry about the offspring yet. What do you know? Huh? Yes and no, because we're dealing with 2 genes, so maybe some of them are, some of them aren't. The parents are the parent generation. So let me. I'm talking like here's parent 1, here's parent 2. What letters do I already know?
Parent 1 is recessive for the yellow apples because it has to be homozygous recessive. And then for the other one, it's small, so it has to be homozygous recessive for the first.
Excellent. Do you guys see what he did there? He said the first one has big yellow. So in order to have yellow, it's got to be little r, right? It's the only way to get yellow. And then the other one's big yellow. So small red. The only way to get small red is to have little a, okay? But let's go back to that. So we figured out the R for one and the A for the other. What's at least some of the A on the first parent?
Capital.
Has to have at least one big.
Got to be at least one big A, right? Now I'll make it easy. We don't know little or big A yet. We'll figure that out in a minute here. So let's leave it at that. What about parent 2? It's definitely got to have one big R. We don't know about the other R yet, right? Because it can be big R, little r, and still be red, because big R is dominant, right? So that's what we've got there. Now it's telling us half and half are big red and big yellow, right? So what would this genotype look like as far as we can deduce? Do your best. Big and red. What does its genotype have to be in order to be big?
Capital R.
Capital A, we're not sure yet, right? What does it have to be in order to be red? Big R, we're not sure yet, right? Could be big R, could be little r. What about in order to be big and yellow? Big and yellow, right? Okay, now, how are we going to get this gene there? Let's start there. What two alleles have to come, what allele has to come from each parent in order to get little r, little r? Little r, right? Well, one of them's coming from here. Where's the other one got to come from? There, right? The only way one of those could be little r, is if both parents have at least one little r, right? All right. Now, we have our big A's, right? And one of them's obviously coming from here, right? And then what are they going to pick up from there? Huh? Little a, right? So that's the only possible combination for the offspring, right? And then this one is going to have to be little r, right? So what are we dealing with there? What letter am I missing?
Big A.
How do you know it's a big A?
There's no small apple offspring.
Yes, we didn't get any little a, little a. There would have been at least one, right? We didn't see any of that. So big A, little r, little a, big R, little r. It takes a little bit of work. Again, you have to be able to logic these things out. But this is very much maybe a bit wordy and a bit long for a test question, but the concept behind this is absolutely something you could see on this test. Okay, so be able to do something like that and end up with that answer.
With that, when it says, like, of the parents, it says, like, I think I should answer my question, actually. The sex-linked genes, is that different than this?
Yeah, so these aren't sex-linked traits because we're not dealing with male or female. Also because they're fruit.
Oh, that's true.
I guess fruit are male and female at the same time. Seeds are technically like the equivalent of an ovary, in case you guys were wondering. Actually, we'll learn that, so you should probably know that eventually, someday. Seeds are the equivalent of a plant ovary, just in case you were wondering. All right. Did anyone have any further questions on that before I move on? Do you just hate that that's even a question? Right? Yeah. Nobody actually cares, in case you were wondering. All right. I do. I care. Sex determination in mammals is due to the SRY gene found on the Y chromosome. Which of the following situations could allow a person with an XX karyotype to develop a male phenotype? Again, pretty sure this was a Mastering Bio question. It's just one that I got emailed to me. So, would it be A, translocation of the SRY to an X chromosome, to a X chromosome? That sounds weird. A person with an extra autosomal chromosome, the loss of the SRY gene from an autosome, a person with one normal and one shortened or deleted X. Okay, who thinks it's A? Who thinks it's B? C? D. All right, pretty much everybody got that one. Why?
Well, because they don't have a Y chromosome, then the translocation switches it to one of the X chromosomes, and then that shows itself.
Yes. So two X chromosomes, you don't have an SRY, sex determining region Y, right? So female is the default sex. But if I have two X's and I want to be, well, don't want to, but in this case, became a male, right? It's because a piece of that SRY, well, the whole SRY, got translocated from a Y chromosome onto an X chromosome, right? If you're having a hard time wrapping your head around that one, try and cross the rest off. How would an autosomal chromosome even affect sex? What is the definition of autosomal? Non-sex chromosome. So that just can't be done, right? The loss of the SRY gene from an autosome, first of all, SRY gene's not even on an autosome, but second of all, losing the SRY gene is not going to make you male, right? It's adding it that makes you male. And then a person with one normal and one shortened or deleted X, what is that? That's just X inactivation. That happens in all females, and they remain female, right? So try, if you're like, I don't know, at least try and cross off as many as you can, right? And with some logic, we were able to cross off the bottom 3, and so even if you didn't know that was the right answer, you know the rest are wrong, so just pick that one, right? But again, it's because a piece of the Y chromosome, the SRY portion, got put onto an X chromosome, and since that's what turns you into a male, even though you have 2 X chromosomes, that SRY gene gets activated and you develop male characteristics. Questions there?
What's X inactivation?
X inactivation is basically you have 2 X chromosomes, but you only need 1 chromosome, right? That's why males only have 1 X chromosome. We still get by just fine, right? So in females of any species, they have the 2 X chromosomes. Well, I guess it has to be mammalian species because they have XX, but they have 2 X chromosomes, so they just turn one off. They just crunch one down like a smushed can, and that means that they have the right dosage. But I have some slides on that because that was another question, so we'll get into that more. Other questions? I know I'm talking like super mega fast, just a lot of stuff to cover. So the faster I talk, the more stuff we get over, right? Or the more chances I have to get off topic. Either one. All right, if cell X enters meiosis and nondisjunction of one chromosome occurs in one of its daughter cells during meiosis 2, how will this affect the gametes at the completion of meiosis? 2 of the 4 gametes descend from cell X will be haploid and 2 will be diploid. All the gametes will be diploid. 1/4 will be N+1 and 1/4 will be N-1. 1/2 will be N. There will be 3 extra gametes. Half of the gametes descended from cell X will be N+1, half will be N-1. All right, who thinks it's A? B? C? D? E? You gave up because everybody else said C? All right, how do we know that?
Because in meiosis 2, it's not taking 4 chromatids, but it's just taking 2 because they've already had independent assortment. So then half of the cells will be just fine, and then the other half will be.
That picture right there is exactly what he just explained. So the left half is if we were to do the non-disjunction in meiosis 1. But this question's asking what happens if we do it in meiosis 2. So meiosis 1, everything runs just fine. And so this half is going to turn out just fine. It's in meiosis 2 that this one doesn't split how it was supposed to in anaphase, and you get that 1/4 N+1, 1/4 N-1, 1/2 N, right? Questions there? Can you go over X chromosome inactivation and genetic linkage from Chapter 15 for clarification? Mostly, how does X chromosome inactivation work? How does genetic linkage work? Can you give an example of what would happen with genetic linkage? So, let's get started. I already kind of explained it, but somebody else explain it in case I did a terrible job of it. What is X chromosome inactivation?
Basically, isn't it when, and it mostly happens in females because they have 2 copies of the X chromosome, where males only have 1 in mammals. But basically, a cell basically says, okay, I don't need to have both these X chromosomes, so I'm going to use this one, and this one's just going to sit there. And with the cat example that's up on the screen, you can get multicolored cats because it's literally like this cell decided to go this way while the other cell decided, I want this instead.
Yes, perfect. So too many X chromosomes in the sense that we only need one, right? So if you're female, you have 2 X chromosomes. So each of your cells individually and completely independent of the other cells says, I only need one, and it just kind of blindfolded, eeny, meeny, miny, mo, let's turn that one off. And that chromosome literally gets crushed. Not crushed, but it gets shortened, right? It gets squished down on itself and physically is unreadable, right? Because we just learned to copy DNA, we open it up and we separate all the strands and we go against lagging strands, leading strands, templates, all that, right? If it's crushed down, proteins can't get inside, okay? So that top cell has the black fur color carrying X crushed, right? And so it's just not going to express black. The bottom one has the orange one crushed. It can't express orange, and so you get this spotting, right? It's called a tortoise shell cat. That's why I kept calling them turtle cats the other time. Somebody pointed out to me this is not the same thing as calico. Calico's black and orange and white. I don't know if that's X chromosome inactivation or what. This specific example is. Question?
So X chromosome inactivation can only occur in females, right?
Yes, if it were to occur in males, you would die. Because you only have one X chromosome, you would have no genes.
So does that mean you couldn't get a male with that kind of color pattern?
As far as I'm aware, no. Because this happens because of X inactivation. So if this cat were to breed with an orange cat, you would either get an orange cat or a black cat.
Okay.
As far as males go. As far as females, you could probably also get another tortoise shell. Yeah. So this will only happen in females. If it were to happen in a male, you would have no sex-linked genes to express, and you just wouldn't. You would die. All right. Other questions?
This gets reversed during reproduction, right? So like during reproduction, the chromosome gets unpacked, and then it can disperse into new gametes, so that like the offspring have the same chance as well. Correct?
I want to say yes. Does anybody? The question is during reproduction, do all of these get opened back up so that they can each get shared? Does anybody remember?
Wouldn't they be just different cells? Like, if it's a gamete, it wouldn't have ever like gotten crushed. So just
Well, I think the question is, so if I were to take that top one and run meiosis so that I could, make more gametes of it, I want to say yes, because if you got the unlucky one with the crushed X chromosome, then you wouldn't get like the X chromosome. If you made a male out of that top one and he happened to have the black one, then he would die or just wouldn't even be born. So I want to say yes, it decondenses, I'm fairly certain, when you form the gametes for reproduction, right? Eggs and sperm. Not 100%, but if I had to take a wild guess, yes. They should open up. Is anyone like 100% sure on that? No? It's embarrassing. I'm pretty sure. We'll go with that. Other questions? All right, so then the other half of that question is, can we talk about genetic linkage, right? And what would happen if you had genetic linkage? So, again, I'm not always the best at explaining everything, or sometimes, just hearing it from a fellow student works a lot better. So somebody else explain to me, in your own words, what is genetic linkage?
So it's when two genes are located really closely on the same chromosome. So if they ever go do like crossing over or anything, they're typically crossed over together, but they're typically inherited together.
Yes, genes are just so physically close together on a chromosome. This picture is terrible because it looks like they're like a quarter chromosome apart, right? I think they just did it so that the B and the VG wouldn't be like on top of each other. Normally they would be like a gene or two apart, if not like right next to each other, right? And so first of all, why can't independent assortment separate those genes?
because they're on the same chromosome.
They're on the same chromosome. Put the left or the right, doesn't really matter. It's It's the same chromosome. You can't separate them by independent assortment if they're the same chromosome. So then that factors in crossing over, right? He mentioned something about crossing over. Why does crossing over not usually separate these genes? Just a really small probability, right? It's a really small target to hit right between two genes right next to each other. It does happen, just not very often, right? So that's why we see kind of skewed numbers, right? This is what we actually see in that cross, because the B VG plus and the B plus VG are considered recombinant, right? Meaning they kind of went against the genetic linkage rule, right? But only 8% of them did that, and the other 8%, obviously, right? The other 42 and 42 stayed how they were, right? Because it's a much higher probability that crossing over doesn't get right between them, and they just stay together.
Does this occur with all genes that are close to each other on a chromosome?
As far as I'm aware, yes. As long as they're next to each other and they're on the same chromosome, they're going to follow this pattern. There may be exceptions, but pretty much a rule. If they're close enough. I don't know what the parameters are. I don't know if it's like, once you're 10 genes apart, it's not going to happen anymore. I don't know if there's even a set number, but they should follow this pattern. So in this case, I want to say this was for like body color being like black or gray, something like that, and then wing type being normal or vestigial, right? So most of the time you ended up seeing, I'm pretty sure it was like black and vestigial and gray and non-vestigial. Could be the other way around, but the principle is the same, right? You saw mostly black vestigial and gray non-vestigial, okay? Because those genes were super close together on a chromosome. The other, I gave an example in my session when I talked about this, about nail patella syndrome, right? And it's where your patella and your elbow bone, I don't know what that is, they grow too big, right? And then you usually don't have fingernails, or you have really, really small fingernails, right? It's a linked gene, right? So these genes for your bone growth and your fingernail growth, if you have nail patella syndrome, are right next to each other on a chromosome. And so they saw people who had the issue with the fingernails also had the issue with their patella, right? In much higher percentages than you should have normally seen, and they were able to deduce, hey, these genes are probably right next to each other on a chromosome then. That's why so often you see both, right? Little or no fingernails and really big kneecaps, okay? Kind of, I guess, a real world example, because none of us work with black vestigial fruit flies or whatever the heck they were. I think they were Drosophila, which I think are fruit flies. I don't know. Nobody even cares, right? Does that make sense? Do you guys have questions still on genetic linkage? All right. So if you were to see these kinds of percentages, that's when you should go, that's linked, right? We're seeing 40%, 40% of the non-recombinant or parental gene, and we're seeing 10% and 10% of the recombinant genes. First of all, if you see the word recombinant, you already know, because we're talking about linked genes. But if it didn't say parental and recombinant and all that, you should still be able to go 40% and 10% is a pretty big gap, right? And so a total of 80 and 20, that's a significant gap. Those are probably linked, okay? All right. I think this was the final one that I got emailed to me. What are the breaks between the lagging DNA strands, and can we go over the differences between leading and lagging strand synthesis? Does anybody remember the answer to that first one? What are the breaks called?
It was the Okazaki fragments.
Okazaki fragments. Some guy named Okazaki found them and said, these are mine, right? It's just the fact that as you go along the lagging strand, you're going against the opening, right? That replication fork, the bubble, right? You're going against the current, I guess you could say. So you have to kind of break it into chunks to be able to keep up, right? Otherwise, you'd start to get pulled backwards. So they just break into chunks, and every time it breaks, you call it an Okazaki fragment. It's a good vocab word to be familiar with. And then can we go over the difference between leading and lagging strand synthesis? No. I'm just kidding. Yes. I have some slides on it, but essentially it's just be able to identify which is the leading strand and which is the lagging strand based on 5' and 3'. But we'll have some practice questions on that. Okay. That's it for emailed in questions.
With the Okazaki fragments, do they really break, or is it just like they just start again and end when they get to the next fragment?
Kind of just breaks down to like semantics. Nothing's like breaking, but it is a stop and a restart. And there is like I guess a gap, and then another protein that I can't remember off the top of my head right now comes in and seals that gap. So there is like an actual space for a small amount of time.
So like doesn't it make it go forward and then jump clear back and make it again in the space? Kind of like.
I'm not sure I understand. Essentially, it's just it goes, and then it does a little bit of a stutter step, and it goes and it stutters, and then after all that, another protein comes back and kind of fixes all the little nicks in it. Does anybody remember what that protein's called?
DNA ligase.
It's the one, whatever it is.
DNA polymerase 1?
Polymerase 1.
Probably. I have some slides on that too, so when we get there, remind me. I can't remember all these proteins off the top of my head. We'll get there though. Questions on that? Okay. Any other questions before I just start going? I should check if anyone's emailed. I do have an emailed in question. Will you explain genetic linkage, please, also with the 42.8% thing? I guess you're referring to this? These two are the parental genotypes, right? Those were the original genotypes seen on the original chromosomes. Because they're linked, because they're so close together on the chromosome, you're going to see a significantly higher portion of them in the offspring, since we obviously don't sort them independently, and they very rarely cross over, and so that's pretty much just the chromosome that they have to get, right? They don't have much of a choice. I don't really know how else to phrase it. If that's still not making sense, maybe go back, watch the lecture. Dr. Pogorski is clearly much better at this than I am, so maybe hearing it from him will help. Read the textbook, do whatever you got to do. Other questions before I get into Friday and Monday's material? Friday and today's material? Can I assume that's a no? It's very impolite not to answer a direct question, just so you know. All right. This is what DNA looks like. Not really, but as far as drawings go. It's made up of a sugar phosphate backbone and then some nitrogenous bases, right? These are our adenine, thymine, cytosine, guanine, right? The nucleotides. Okay, the A, T, C, and G that you see all over the place, okay? And it always runs 5' to 3', okay? In this case, 5' to 3'. All right? It boils down to where OH is and where phosphates and all that are on, but you don't need to know that so much as know it goes 5 to 3, always, okay? You won't be like shown this picture without like the 5' and 3' label and say, which one's 5' and which one's 3'. You'll be shown two fragments. One of them will be labeled to a certain degree, and you'll have to like figure out the rest of them, okay? And I have some questions like that. Questions on DNA structure? So Chargaff's rule is just that the amount of A always has to equal, within a very close percentage, the amount of T, and then G always equals concentration of C, right? So just AT, CG. Memorize that. I don't know how you want to. I know the only mnemonic I know is apple tree chewing gum. That doesn't help make up your own. Whatever, right? But memorize that rule. And it is a rule. We don't break this, right? You may break it by like 0.1% or something, but you won't break it enough.
So if you have, like, say, 19% C And then how do you find like how much you have of A?
Perfect. So if we have the concentration of C is 19%, what's the concentration of A?
Yes.
Who wants to help me out?
You can just take 50 minus 19.
Take what?
50, since we know that C is going to be equal to G, so we know that the remaining percentage will be equal. But like that and A will take up 50% of the molecules that cells.
Not 50%, because it's not like an even, this is 50 and this is 50. You're close.
So you can just take 19% and multiply it by 2, because we know C and G are equal. And then we take 100 minus 38, and then divide that by 2. It's probably a horrible way to do it, but
Did I do my math right? Pretty cool, right? I was like, I don't know if that's 38, I'm pretty sure. Yes, so take your 19 that you get from C, and we know the amount of C has to equal the amount of G, right? So really we have 38% of C and G. So we have to have 100% total. We take out the 38 made up by C and G, will tell us how much we have of A and T. So we have 62% A and T. But we just want to know how much A we have, so divide that in half, since A and T are an equal proportion. So we have 31% A, which actually also happens to be the amount of T. Does that help? And I actually have another question on that, so we'll see it with different numbers, right? Because I know it's you hear it one time and you're like, okay, yeah, I see that, but then you see it with different numbers and you're like, ah. So I have another one. I saw a hand.
I just actually wanted to say that the 50% that he actually proposed would work because C plus A and G plus T, those both have to equal 50% at some point.
Oh, I see what you're saying. So like that had to be 50.
Yes.
Take an opposite base pair minus
Okay. I've literally never seen anyone do it that way before, so I apologize. That did work. I was like, I don't think you can do that. I've never done that. So whichever way works for you. If you want to do like 50 minus the amount, and then that just tells you whatever works, whichever one's easier for you. If I ever saw this on a test, I would do it this way, only because that's how I've learned it. But if this makes more sense to you, do it that way. I would only say, don't risk it for the biscuit and try and like do it a faster way that you don't really know how to do. If the way you know how to do it is slower, do that one, because that's how you know how to do it, right? So whichever one makes more sense to you, go with that. My way's better. It's actually not. It's slower, but it works. All right. If a DNA molecule contains 15% of its bases as C, then the percentage of bases that are A is, take a couple seconds to do some mental math. All right, who thinks it's A? B? C? D? E? All right, an overwhelming percentage of you said D. Somebody walk me through what you did. Don't use the 50% rule. No, I'm just kidding. Do whichever one you like. Right here?
Okay. So you have 50% C, so that means there's going to be 15% G bases. So that gives a grand total of 30% of all the bases. So that means your A and T bases have to equal 70%. And then you're going to divide that by 2 because you have 2 bases, and you get 35% on each base.
Boom. Right? Does everybody see how that's working? I'm not even going to try and do the 50% way. Cool?
Is that also for T?
Yeah, so that also, had it asked you T, it would have been the same thing, because T and A equal each other. If it asked you what's the concentration of C, what would have been the answer? 15, right? It's not going to be that easy on the test, but it's a good way to tell whether or not you know really how this is working, okay? All right, so hydrogen bonds are what hold our DNA together, right? Just another reason hydrogen bonding is super important. Each strand is the template, right? So we take this strand and we break it in half. That was ugly. Okay, and we copy against that left side to make a whole chromosome. We copy against the right side to make a whole chromosome. And notice how that one and that one are exact copies of that one. Okay? So you may go, well, the left half and the right half aren't the same as each other, so how are you going to get the same chromosome? You use the left half as one template and the right half as the other template. And since they're complementary to each other, that ACTAG is going to make TGATC, and then the TGATC is going to make ACTAG, right? So they work as inverse templates of each other, I guess. I don't know. That's like math terms, but I'm not in math anymore, so I don't know. I gave up after calculus. Does everybody understand what I'm saying? Get a lot of blank stares here. You guys just bored? Is this just too easy? Should I make it harder? Bidirectional replication from origins of replication. What the heck does that mean?
So, yeah, in your bubble you have basically they're going opposite directions. So they're just opposite force basically. One goes that way, the other way.
Perfect. So we take two origins of replication, right? One on each strand. We open up a bubble and we replicate bidirectionally. We open up several of them, right? Makes it go a lot faster. Bacteria just do unidirectional replication because their DNA is super short, so they literally just kind of like open one end and go zip, and they're done. Our DNA is significantly longer, so we open up all kinds of spots all along a chromosome, and they end up kind of running into each other, right? So if you open up 10 replication forks, I guess 20 replication forks, 10 bubbles with 10 replication. 20. You get what I'm saying. 10 bubbles, 20 replication forks. It's going, I don't know, 20 times faster? I don't know how much faster. A lot faster. You don't need to know how much faster, just that it's faster. I feel like I said faster 400 times. You guys are struggling today.
Could you go over, I mean, I know it goes 5 to 3, but it gets really confusing when you've already got your parental strand is 5 to 3.
Yes, I totally get what you're saying. It's the worst. I don't understand it either. No, I'm just kidding. You guys are like, frick, we're all going to fail. You might. You definitely might. No, that's terrible. That's terrible. I got to get you guys to react somehow. Yes, I have a couple of practice questions where we will work through how to decide who's 5, who's 3, which way is leading, which way is lagging, where is it going, all kinds of stuff. Good stuff. Those are very much test type questions, so be very good at that. All right, DNA synthesis requires many proteins, right? So it's a wonderful blanket statement. It requires more than one protein. Great. Let's actually go through what they are, right? Hopefully you studied up on these. If not, this will be a good chance for you to, I guess, test yourself on how well you already know these, because guess what? You're going to have to know which ones are which. So some of them I've covered their function, and some of them I've covered their name. So either know the function of helicase or know, which protein binds to and stabilizes single-stranded DNA until it is used as a template, right? So let's start. What does helicase do? I heard literally one person answer me.
Doesn't it separate the two parent strands? I don't know.
Yep. It unwinds the parental double helix at replication forks. So basically, you have your DNA strand stuck together, and the helicase jams itself in there and opens it up, right? Like zippers on pants, right? That's why I made the joke last year about helicase being the sexiest enzyme, because it unzips your jeans. Got some backlash for that. I clearly don't care because it's like the third time I've said it this semester. Say what you want. You're not my boss. Yes, it opens your genes. It unzips your genes, right? And so if the helicase joke helps you remember that, then go with that. All right. Which protein binds to and stabilizes single-stranded DNA until it is used as a template? I heard like 3 people mumble it. Single-strand binding proteins, right? Bind, strand. I guess none of them say protein. Oh yeah, single-strand. Single-strand binding protein binds to a single strand, okay, until it's ready to be used. Okay? That's just to keep it from like closing back on itself, right? So it's like, I can't think of a real world example of this now. Anybody help me out? Something that keeps it from closing back. What?
It's like a zipper jam, and you can't close your coat when it's freaking cold outside.
The quote that
It'd be like if you had a piece of gum in your zipper, and you're like trying to zip your jacket up, but there's like gum right here, and you're like, yeah, that works. I guess. All right. What does topoisomerase do?
It cuts the DNA so that it can unwind the twists.
Oh. Relieves overwinding strain ahead of replication forks by breaking, swiveling, and rejoining DNA strands. A very fancy way of saying exactly what she just said. It cuts the DNA so that it doesn't get too tight, right? Dr. Pogorski uses the example if you had like 2 ropes, right, all wound together in a sort of a double helix, right, and you took each end and you did this, you would only be able to open it so far before the rope would get what's called supercoiled, and it's so like tensed up on itself that it's not going to open any further, right? So a topoisomerase comes, snips that, and you do it again, snip, do it again, snip, do it again, right? It's the whole rule.
So does that just move down a strand, or are there multiple?
I'm sure there are tons of them because the strand is super long and you have tons of replication forks, so it's going to be along the whole thing. I don't know how many, I don't know what like the portion would be, but yeah, but it's tons of them. All right, which protein synthesizes an RNA primer at the 5' end of leading strand and at 5' end of each Okazaki fragment of lagging strand? Huh? Primase. You're right. It's primase. So it synthesizes an RNA primer. Okay, ACE just means enzyme, right? Enzymes are proteins. So this is a protein/enzyme that puts down a primer. What does a primer do?
has the bases on the other side. It primes. It literally primes the DNA strand for more bases to be added on.
Yeah, you guys ever heard of priming a pump? Putting primer in a
Priming the lawnmower.
priming a lawnmower, right? You push the little bubble thing 500 times till it puts like a little bit of oil in there, and you're right. So it just gets things started. Okay, so a primase puts down a primer on the 5' end of the leading strand and the 5' end of the Okazaki fragment on a lagging strand, right? Because which way does synthesis go? 5 to 3. So where should we put a primer? On the 5 end, because we're going to go toward 3, right? If you put it on the 3' end, there's nowhere to go from there, right? So primase puts down primers. All right, what does DNA polymerase 3 do?
DNA synthesis.
Synthesizes new DNA strand from the parental strand by adding nucleotides to an RNA primer or an existing DNA strand, right? So it's sticking the nucleotides on there, okay? It's the guy doing the actual heavy lifting, right? Someone actually has to put nucleotides against the other nucleotides, right? We can't all just unwind and prime. Someone has to actually synthesize. So what does DNA polymerase 1 do then? removes RNA nucleotides of primer from 5' end and replaces them with DNA nucleotides, right? So remember, our primase put down RNA primers, right? But we don't want RNA in our DNA because we just want DNA, right? So then polymerase 1 comes back and says, okay, you're good. Now we want this to be DNA, and it swaps out so that they're DNA nucleotides instead of RNA. Okay? The last one, DNA ligase, what does that do? Joins Okazaki fragments. So this is the protein that I couldn't remember off the top of my head. Right? We make all those Okazaki fragments. We got little gaps between. So then our DNA ligase comes, and it fixes all the holes, right? So that we have one continuous new strand of DNA. All right? Memorize all, 7 of those. Know their functions well enough that if you were asked, if you had a genetic defect where single-strand binding proteins no longer perform their proper function, what would happen? And you would have to be able to say, you wouldn't be able to replicate your DNA because you would open it up and it would close back on itself, right? And nothing is there to keep it from closing back on itself. Or what would happen if you had a mutation where DNA polymerase 1 no longer works, right? You would no longer be able to take the RNA nucleotides and switch them over to DNA, so you would still have RNA stuck in your DNA. Okay? That's a test type question. If you had a mutation that made such and such protein stop working or work too hard or something like that, what would happen? Dr. Pogorsky loves the hypothetical, if a mutation screwed up this, what would happen? So maybe ask yourself those kinds of questions as you go through, right? Questions up to there?
So would an excess of, say, DNA polymerase 1, would that really affect it?
Probably not. On that one, I would say no. As a matter of fact, I don't know if an excess in any of these would really do anything. Yeah, so as far as these go, you would probably see more of a if they stopped working type question. But in general, Dr. Pogorsky loves to say if it stopped working, if it overworks, if it, just those kinds of things in general. But yeah, with these, you probably only see if it stopped working. Other questions? Oh. DNA strands are anti-parallel, meaning that the 5' to 3' is opposite in the strand against it, right? 5' to 3' down, 5' to 3' up. Okay? That helps us when we get a question about something like this, right? And it says, which is the leading strand, which is the lagging strand. If you weren't shown, first of all, the labels, but second of all the gaps in there, how would you know? How do you know which one's which?
If we look at the arrow, the red arrow in the replication fork, it's going towards the fork.
The one going into the fork is the leading strand, right? The one coming out of the fork is the lagging strand. What if you didn't even have red arrows in there?
It's going to start at the one with the 3 because it starts right. Yeah. So because it's opposite, so it's going from 5' to 3'. So it'll start at the 3' of the replication fork.
Yeah. So if this one is running 5' to 3', so the gray parental strand is running this way, then the red copied strand has to run the opposite direction, right? It has to run, so this has to be 5, and this has to be 3. So I find that for me, the easiest thing to do, if I'm not given that number, if I'm not given any of these in here, right? In fact, you could even do it if you literally only had one number. If you just had that 3' down there, you would go, well, if I know that's 3', so that's got to be 5. So the strand next to it has to be 3. So this end has to be 5. So the strand next to it has to be 3. And the strand next to this one has to be 5. So we're obviously going 5 to 3 and 5 to 3. So the strand going in is leading, right? If you're like, I still don't understand how to do that, let's practice it, okay? I've only given you, these are the two parental strands, right? I've only given you two numbers. That bottom one, bottom left, is 5' and top left is 3'. Which strand would allow DNA synthesis to move left to right? Upper strand, lower strand, both strands, neither strand, I don't care. Take a second, maybe draw it. Use your hands. I see a bunch of you doing that. Nothing wrong with that. People in the testing center will be like, what? It works. What do you care, right? If you get an A on the test, doesn't matter if people think you're using sign language. Well, unless the proctors think you're using sign language. Then they're going to be like, who are you talking to? Talking to the biology gods. All right, who thinks it's the upper strand? Who thinks it's the lower strand? Who thinks it's both strands? Please never answer both strands. You cannot have a leading and a leading strand. It just doesn't work like that. And obviously it's not going to be neither strand, right? And hopefully you care, because if you don't, you're in trouble. It is the upper strand. Somebody walk me through how you found that answer. So it has to go 5' to 3'. So maybe I circled the wrong one. Oh, maybe it didn't format over right. Hang on. No, we're doing this wrong. This is the 3' end. This is the 5' end. So this is the 3' end. And so the complementary strand, oh no, we're doing it right. I'm getting myself confused. It's going 5', it's going left to right. I was doing right to left. I don't know what I was thinking. So yes, if this is the 3' end, then the complementary strand has to be 5', and we always go 5 to 3. So that top strand is going left to right. Does everybody see how that happened? Let's try another one. Different question to test your vocab knowledge. Which template would have the leading strand? Upper, lower, both, neither, I don't care. Again, please cross off those bottom three immediately. You know it can't be both. You know it can't be neither. You're not allowed to answer, I don't care. Who thinks it's the upper strand? Who thinks it's the lower strand? It's got to be the upper strand, right? It's literally the same problem. It's 5' right here, and we're going the same way. The leading strand is always the one leading into the replication fork. And the lagging strand is the one coming out of the replication fork, because this is, right, 3'. And so we're kind of going against the direction that we want to, so it's going to lag, right? You guys see that? Same question, different way of phrasing it. Which one's going left to right, or which one is leading and lagging, right? So on the previous question, had it asked which would allow DNA synthesis to move right to left, then it would have been the lower strand, right? Because now we're going right to left. Or if it had asked which way allows us to go, or sorry, which one's the lagging strand, then it would have been the lower strand, right? So he can phrase it however he wants. He can't trick you as long as you just find which one's 5', which one's 3', right? So just always go, well, if that's 3, that's 5, and if that's 5, that's 3. So which way am I going? And done, right? Does everybody see that? All right. Oh, this one didn't format over. That's not good. Shoot. I believe it was more like, oh, shoot. It's supposed to be right there. Okay, that should work. Okay. Consider the following primer template pair, right? We have the actual DNA, 5' to 3' is AAA, CCG, AGT, ATTG, right? And then we're copying that top strand. We already have a GGC, TCAT, okay? So what is going to follow? Would you have an AAG an AAC, a GTT, a TTT, or a CTG. Okay? Take a second, orient yourself, figure out, they've got to be complementary, right? So if it's a T, it's got to be an A. If it's a G, it's got to be a C, vice versa, right? Maybe write it down, draw it out. What do you guys think? A, B, C, D, E. I've fooled you. Most of you said it was B, right? Because you were probably going based off of this, right? But we don't go that way. Because this is 5' and this is 3', so you're going this way. I tricked you because you read left to right. No? And so you probably just went GGC, TCAT. All right, so then it's got to be AAC. Pay very specific attention to which way you're running, okay? It doesn't read left to right as a rule. It can read left to right or it can read right to left. So it's TTT because you were trying to fill that space right there. I know this formatted over weird. This was supposed to be like right there. Okay, you guys see that? All right. Way over time. People are waiting out in the hall, so we got to call it. Sorry.